We just learned that 3.5 billion years ago, abiotic molecular evolution happened, and through that, protocells were formed. With the absence of oxygen in the atmosphere, there was the formation of organic molecules like proteins, carbohydrates, lipids, and nucleic acids. With the help of external energy sources such as lightning, heat, UV rays, or UV light. Um, the protozoals were formed from these organic molecules, eventually becoming the living cells that we have now. And they are capable of reproduction and metabolizing food. This happened because of the evolution through natural selection. Now, we look into the details of the cells. Now, we want to know what are the cell compositions in this discussion.
Cells are classified into two different types, the prokaryotic and eukaryotic cells. Prokaryotic cells evolved earlier, which explains their simple structures. There is this hypothesis we are all familiar with, the endosymbiotic hypothesis, wherein the ancestral cells engulfed the prokaryotic cells and had a symbiotic relationship. These prokaryotic cells eventually evolved into the organelles of the eukaryotic cells. Now, in this illustration, it shows the similarities and differences in structures of prokaryotic cell and a eukaryotic cell. Both types of cells have plasma membrane, cytoplasm, a DNA, or a region where the genetic materials are concentrated, and ribosomes. The main difference is that the eukaryotic cells have membrane-bound organelles, which means that the intercellular structure are surrounded with layers of organic compounds. There are different groups that are classified under these types of cells. Um, all living organisms are grouped into three domains. The prokaryotic organisms are classified under domain bacteria uh, and domain archaebacteria. And for all eukaryotic organisms, they are grouped into one domain, which is the domain Eukarya. So this is the domain Eukarya. Under that are kingdom Animalia, Plantae, fungi, and Protista. We also have domain Archaebacteria and domain Eubacteria. So let us further compare prokaryotic and eukaryotic cells. Um, prokaryotes means before nucleus and eukaryotic means after nucleus. In the previous illustration, we saw that the prokaryotic cells only have a region where the genetic materials are located, so there is no nucleus in the cells, compared with eukaryotic cells with a true nucleus, a membrane-bound organelle. Eukaryotic cells were formed 1.5 million years ago, while prokaryotic cells were formed much earlier, 3.5 billion years ago. In terms of size, prokaryotes are very small, while eukaryotes have larger cells. All prokaryotes are um, unicellular and less complex, and most eukaryotes are multicellular and are capable of complex processes. Also, prokaryotes reproduce asexually through binary fission and can survive in an anaerobic environment, so without oxygen. Eukaryotes, meanwhile, can reproduce both asexually and sexually and utilize oxygen in their functions. So this is just to show you the prokaryotic cells or what prokaryotic cells look like. They usually have cilia and flagella that are used in different functions such as locomotion or movement, and some have don't have cilia or flagella. There are different ways that prokaryotes can be utilized. They are called workhorses because of these. Um, they are small, but they can multiply really fast. They are usually used in genetic engineering, such as the E. coli. And an example is the industrial enzyme produced by organisms under archaea. The enzymes are suited for a wide range of temperature, pH, and ionic strength. Again, just to show you the difference in structures of the prokaryotic and eukaryotic cells. So for prokaryotic cells, we only have this region of where the genetic materials are concentrated. But for eukaryotic cells, we have nucleus, which are membrane-bound organelles, and other organelles that also have membranes. Here are the different kingdoms formed because of evolution and diversification of prokaryotes and eukaryotes. So prokaryotic organisms were grouped into eubacteria and archaebacteria. So eubacteria are unicellular and of course prokaryotic organisms. They differ chemically from archaebacteria. So archaebacteria are also prokaryotes that can live in extreme environments. So for eukaryotic organisms, we have plants, animals, fungus or fungi, and also protists, the unicellular eukaryotic organisms. So just a quick look at eukaryotic cells. So in eukaryotic cells have these structures. You have organelles or sub cellular structures that has one or more specific jobs to perform in the cells. They are usually surrounded by a membrane. They also have uh, cell wall or cell membrane, cell wall which is present in plants and also in fungus or in fungi. And also we have endomembrane system, which is a membranous structures which is involved in the transport within the cell. Um, includes endoplasmic reticulum, Golgi apparatus, and um, cell membrane, nuclear envelope, and the vesicles. So we go back uh, to microorganisms. So all prokaryotes are considered microorganisms. However, eukaryotes also have representatives that are single-celled microorganisms, and they are included in the group called protists. So prokaryotes, we have two domains here, two groups. You have archaebacteria and eubacteria. And protists are eukaryotic organisms that are unicellular. So again, here are the six kingdoms that uh, you should be familiar with. Animalia, Plantae, Fungi, and Protista, which is under eukaryotes. And the two groups that are considered uh, groups of prokaryotic organisms. We have archaea and archaebacteria and bacteria. Let's talk more about prokaryotes. We know you know now that uh, they are the first organisms that inhabit Earth, and through their long evolutionary history, the prokaryotic cells were subjected to natural selection in all kinds of environments, resulting in their enormous diversity today. So they're found everywhere, including in and on the bodies of multicellular organisms. The collective biological mass or the biomass of prokaryotes is at least 10 times that of all eukaryotes. So one key feature of prokaryotic cells is the cell wall, which maintains the cell shape, protects the cells, and prevent uh, or protect the cells from harsh conditions. The cell wall of prokaryotes is different from those of the eukaryotic cells, such as plants and fungi. For the cell walls of plants, it is composed mainly of cellulose and chitin, while for prokaryotes, the cell walls contains peptidoglycan, which is a polymer composed of modified sugar com- um, linked by a short polypeptide bond. And because of this property, scientists can categorize different bacterial species based on the difference of their cell wall composition. And the technique used is the gram staining or gram stain. The bacteria that have cell walls with large amounts of peptidoglycan are called gram-positive, while bacteria with less peptidoglycan are called gram-negative. As you can see in the illustration, the colony of bacteria uh, was stained with here. And also bacteria are also diverse and well spread, and in fact, just in the human body, we can isolate several species of bacteria. And if you look at this illustration, we can see that in our palm and in forearm, we can have diverse uh, species of bacteria. So these are the 10 trillion friends that you didn't know you had. So there are also different sizes and shape of bacterial cells. The average size of a bacteria is about zero point 0.2 to 1 micrometer, and the basic shapes are spherical, rod-shaped, and spiral. So for spiral or for spherical, we call it coccus. For rod shape, it is a bacillus. For spiral, we have a spirochete or vibrio, spirillum. There are also unusual shapes like star-shaped, also called stella, and there is also square-shaped bacterial cells. Most bacteria are monomorphic, which means they exist only in one form, while there are also few bacteria that are pleomorphic, which means that they have uh, irregular and variant forms. So, same species will have um, different forms in terms of shape. Here are the common shapes of prokaryotes. You have the coccus or the spherical prokaryotes that may occur in pair, so we call it diplococcus, or in chains, which is called streptococcus, and in clusters, or staphylococcus. There's also bacilli, or rod-shaped prokaryotes. They are usually solitary that may also form chains, so we call it streptobacilli. There are also spiral prokaryotes, which include spirilla. They have various forms, such as a comma-like shape, loose coils, or cork shape like the spirochetes. So there are two domains of prokaryotic organisms, bacteria and archaea. Archaea are prokaryotic prokaryotes that inhabit extreme environments, and of course, we are all familiar with bacteria. Here is the comparison of the characteristics of archaea and eubacteria. Both have uh, no true nucleus. For eubacteria, they are heterotrophs or autotrophs, and for archaea, they are all heterotrophs. The cell wall of eubacteria is composed of peptidoglycan. Peptidoglycan is absent in the cell wall of archaea. The eubacteria live can live anywhere, everywhere. For archaea, they live in extreme environments, such as environment with very high salinity, we call it halophiles, or with very high temperature, thermophiles, or with a large amount of methane or methanogens. So microbiologists analyze the genetic compositions of the prokaryotes, and the genetic diversity of prokaryotes are immense. Here are some examples. You have actinomycetes, cyanobacteria, and this species of Theomargarita, which is known as one of the largest bacterial species in the world. So here is the simplified phylogeny of prokaryotes. This phylogenetic tree is based on molecular data. So from the universal ancestor, it diverged into different groups. So you have the eukaryotic organisms and also or the prokaryotic organisms here, the archaea and the bacteria. So for bacteria, we have different groups under it. You have the proteobacteria, you have the chlamydias, the spirochetes, the cyanobacteria, and the gram-positive bacteria. So for proteobacteria, we have several subgroups. We have the alpha, beta, gamma, delta, and epsilon proteobacteria. Hmm. So for alpha proteobacteria, we have representative species called the rhizobium, which can be found inside the root cell of a legume. So many species of in this subgroup are closely associated with eukaryotic hosts. For example, this Rhizobium species live in the nodules within the roots of legumes, and the bacteria converts atmospheric nitrogen to compounds that host plant can used to make proteins. For beta proteobacteria, um, this nutritionally diverse subgroup includes Nitrosomonas, a genus of soil bacteria that play an important role in nitrogen recycling by oxidizing ammonium, producing nitrite as waste product. Also, another subgroup is the gamma proteobacteria. These subgroups autotrophic members include sulfur bacteria such as Theomargarita, and which obtain which obtain energy by oxidizing sulfuric acid, producing sulfur as the waste product. And some heterotrophic gamma proteobacteria are pathogens. For example, Salmonella, which is responsible for some cases of food poisoning, and Vibrio cholerae causes cholera. And under this subgroup also includes the E. coli or the Escherichia coli, which is a common resident of the intestines of humans and other animals, which are normally is not pathogenic. Another subgroup is the delta proteobacteria. This subgroup includes um, the slime secreting myxobacteria. When the soil dries out or the food is scarce, the cells congregate into a fruiting body that releases resistant myxospores. And lastly, the group of epsilon proteobacteria, and most species in this subgroup are pathogenic to humans or other animals. Epsilon proteobacteria include uh, Campylobacter, which causes blood poisoning and intestinal inflammation, and also the genus Helicobacter, which causes stomach ulcers. Now we have this specific bacterial disease called the necrotizing fasciitis, which is caused by a bacteria called Streptococcus pyogenes. It can destroy skin, fat, and tissue covering the muscles within a short period of time. But also, bacteria can be very beneficial, specifically for humans. So humans can use bacteria to improve healthcare. They can use also for fuels, for the industry, and for agri-tech and the environment. So it can help clean up oil spills, for example. They can also produce antibiotics. Um, it can also be used as food supplements, and bacteria can convert waste into fuel. They can also break down uh, different um, organic compounds, and it can use for the production of certain foods such as cheese and yogurt. And one specific use of bacteria is for bioremediation, which is the use of these organisms to remove pollutants from water, air, and soil. And the prokaryotic decomposers is used to treat sewage, and this this can also be used for oil spill and also for the treatment of polluted water and soil. So an example of the process is that they inject the microbes into the polluted water, and the microbes utilizes they break down the contaminants with the help of the enzymes, and as a product they release carbon dioxide and water.